So the talks have been really exciting. The robotic lobster, I think, was really cool. So I want to thank you for that talk. That was really amazing. All right. So what I'm going to talk about is our attempt to create a synthetic form of yeast with entirely lab-created DNA. I'll start with credits for the funding from Autodesk. So I hadn't really known about Autodesk before we started this project, but Autodesk is one of the main makers of CAD/CAM software. So designing things like buildings, cars, desks, and they also do movie animation. And they think that synthetic biology is going to be a cool new way of making things, and so they want to get into the area of doing CAD/CAM for biological systems. And then DARPA, so actually we just heard from the robot talk and from previous talks, DARPA, so this is the American Defense Advanced Research Project, is really big into synthetic biology. DOE, we've gotten funding from the Department of Energy. They're interested in energy applications. Microsoft Research, surprisingly, has a big effort in synthetic biology, and National Science Foundation. So the one agency that is conspicuously missing here for US researchers is NIH. NIH really hasn't really seen much use for synthetic biology yet, but hopefully that will change. And I think talks like the previous one today about synthetic biology applications in cell and tissue engineering are really going to be cool and important. So that's one possible area for future work. All right, so this is to sort of situate the research that you'll be hearing about from me. So this is looking sort of the landscape of synthetic biology. On this axis, size of nucleotides, and on this axis, the type of target. There's a lot of exciting work in improved synthetic methods for small pieces of DNA. We've heard a little bit about protein engineering. A lot of work that people hear about and talk about is sort of the level of 10,000 to 100,000 nucleotides of engineering pathways, so iGEM type stuff. But what we're talking about is synthetic genomes. So 1 million is sort of small. We're getting, I guess, by the end of our project, hopefully 12 million base pairs synthetic life. And sort of in analogy with what people have been doing or did at the start of organic chemistry, synthesizing organic natural products, we're starting with living natural products that are genomes. So that's what our targets are. So why do we want to synthesize something that already occurs in nature or is sort of similar to something that occurs in nature? So this is a famous synthetic biology quote from Richard Feynman's Blackboard. Although he was talking about engineering, what I cannot create, I do not understand. There's this other quote right underneath it that he wants to know how to solve every problem that has been solved. But so it turns out that he was sort of a lazy guy in that when he got a problem, instead of going to the library to see if it had been solved, he would try to think about it and solve it on his own. So a lot of the problems he solved, they're actually known solutions, but it's faster for him to just work out the details himself than to go to the library. So maybe that's a bit like us with synthetic biology, that we want to get to the point that if there's some DNA sequence you want to make, instead of cloning it out of a cell that expresses it, maybe it's faster just to type it in and then order it up from your sort of integrated computer synthesis environment. All right, and so finally, something that's motivating our project and others is that we finally have the technology to do synthetic biology large scale. So this is a graph showing progress of different technologies. So this is year, and this is sort of efficiency. This is Moore's law. And then these points are DNA sequencing, which is going up faster than Moore's law. And I stopped this in 2003 because that's when the 454 genome sequencer was the fastest genome sequencer. And I helped invent that, so I'm still sort of proud of this. But now we're left in the dust by other platforms that are really sort of going faster and faster. And in fact, it's getting to the point, so you see this is going up faster than Moore's law. What that means is that it's getting to the point where it's cheaper to redo a whole genome, to redo a genome sequence than it is to pay the cost of storing the data on your computer. That if you look at, say, what it costs on Amazon, $1,000 per terabyte year to store your data, it's less than $1,000 per terabyte to generate DNA data. So it's a real challenge to figure out what to do. And then the circles here are DNA synthesis technologies that are also going up very fast. All right, so that brings us to the projects that are feasible. So it's really feasible now to make all the DNA to run a cell. So this is a project going on at Johns Hopkins. I'm doing the computational bioinformatics stuff. Jeff Buca is, I guess, I'd say overall project leader, the experimental side. Srinivasan Chandrasegaran is an expert in the technologies to assemble large pieces of DNA. One of his previous projects was doing zinc finger-coupled endonucleases. So these are the products where the hope is that if you have a mutation in your DNA, you can engineer a nuclease that one part of it is engineered to recognize the DNA sequence, and the other part is engineered to cut it out and do DNA surgery. So, so we're using some of those similar technologies to put together pieces of DNA. And then people who've been working on the stuff I'll show you, Giovanni Stracquadanio, an optimization PhD who's doing a postdoc with me, Patrick Kai, who's working with Jeff, Jessica Diamond and Sarah Richardson were graduate students. Sarah's now at LBL Joint Bioenergy Institute. We've also internationalized this project. We have collaborators at Tsinghua and Beijing Genomics Institute, and I should have mentioned Tom, I'm sorry. So now Tom Ellis also, we're branching out to Britain. All right. So, all right, so our target is Saccharomyces cerevisiae. So version 1.0 is the naturally occurring yeast. So if you're going to do a project in eukaryotes, yeast is the obvious first organism to do. It's the best characterized, and it has the best reagents for doing any sort of molecular biology or genetics. It has 16 chromosomes compared to human 46 chromosomes. 14 million base pairs of DNA as opposed to human 6 billion base pairs of DNA, diploid versus haploid. So about 6,000 genes versus humans 20,000 genes. It makes interesting stuff. So it makes beer and bread. It's also used a lot in industrial processes. And, but basically, it's the clear first organism to go to if you're doing any sort of new type of project. And a lot of synthetic biology has been done in E. coli, not as many tools in yeast. So part of this is also the idea of bringing tools that have been developed for prokaryotes to use in eukaryotes. So, so we're not synthesizing an exact copy of the yeast reference genome. We're making a lot of changes all over. So, so three main goals of what we've been doing. So first, so Google would say don't be evil, or a doctor would say do no harm. But so we want to maintain, sort of over the short term, we want to maintain wild type growth. That the changes that we make, we don't want to be a big burden to the cell. Over the long term, we want to maintain genome stability. So we want to do things like removing junk DNA, like transposons, not really junk, but transposons are genetic elements that have invaded eukaryotic cells and reproduced. And then they've been tamed by eukaryotic genomes, but the transposons are still there. They haven't been selected out yet. But so our hypothesis is that if we get rid of these, they're serving no beneficial purpose to the cell. Maybe the cells could grow a little better. But more importantly, transposons, sometimes big long repeats, are sites of genome instability where genomes can fragment or recombine in ways that you don't want. So, so we want to see what will happen when we get rid of those. And we also want to increase the genetic flexibility of the cell. So things like codon removal to open up a codon to put in natural amino acids, cleaning up the restriction enzyme sites so that you can do easier restriction ligation genome scale. And then we also are making the genome in a way that allows it to evolve very rapidly. Through LOXP sites. So this is sort of a restatement of the previous slide. So another reason, well, yeah. So a little bit more on the sequences that we're removing. So removing transposons. We're also removing introns. So there are a lot of hypotheses about what introns are for. In human, there's alternative splicing. In yeast, there's not really so much alternative splicing. And so it seems that yeast doesn't really, in terms of protein products, it doesn't really use introns. So if we get rid of all the introns, can we also get rid of RNA splicing? There are hypotheses about how RNA splicing is linked to translation. But again, in yeast, since most proteins aren't spliced, it seems like maybe you could entirely get rid of RNA splicing. So there are huge parts of eukaryotic biology that you can only try to do experiments with if you have a synthetic species that, say, doesn't have any introns. Also, one thing that we're doing is removing all of the tRNAs to their own chromosome. So we're making chromosomes that have specific functions. And the motivation for that is that tRNAs are sites of genome instability because they are transcribed heavily, and that makes single-stranded DNA, and single-stranded DNA is more likely to break or get errors. So, so that's what we're going to. So this is the one ethics slide. It's very important to talk about ethics when you talk about synthetic biology. But it turns out that, so at least in my experience, people have been talking about the same thing in ethics for maybe at least 100 years, maybe more like 200 years, at least back to Frankenstein, or Frankenstein's monster. So there are three things that worry people, things that can propagate, things that are intelligent, and things that can evolve. So since those robots that we heard about, they don't really learn, and they can't propagate, and it sounds like they're not so intelligent. So maybe those robots, so people aren't going to be worried about the robo-lobsters, unless they're sort of crawling into your house and listening to your email. But so things that can propagate, so like zombies and nanobots, they propagate, but they're not intelligent, and they don't evolve. Synbio is sort of between propagating and evolving, but not so intelligent. Vampires are sort of up here, and then aliens are in the center. They can do everything. Artificial intelligence, it's more intelligent and it can evolve, but I don't know, maybe propagation by taking over all the computers. So actually what's missing here, and I think is actually sort of missing from what people talk about with bioethics, is there's no axis for evil on this. I mean, so I think maybe people in ethics aren't so who knows what evil anyway. And then so this is, so if you haven't seen the movie, Hunger Games was a very enjoyable movie. I was a very bad parent and took my 10-year-old and 12-year-old to it. All the other parents in our community were very upset with me for being a bad example of letting our kids see a troubling movie. But so spay and neuter mutations, so essentially so they don't propagate. All right, and then so orthogonal life, to try to make our lab strain orthogonal from sort of wild yeast, we are starting to work with Theron Isaacs at Yale, who's done recoli to sort of spin the codon wheel to recode a stop codon for an existing or a new amino acid. So even doing something like recoding a stop codon for an existing amino acid would mean that then if your cell got out, then and it traded DNA with another cell, the things that it made wouldn't be functional. So there's a lot of interest in doing this for adding new functionality, but also for insulating or making your organic stuff, making your synthetic life orthogonal from other life. So so that's the motivation. Now here are results. So this is our design for the first two synthetic chromosomes we've made, actually two synthetic chromosome arms. So this is 6, I think, and this is the right arm of 9. And so what these show are each of these symbols is a yeast gene, an open reading frame, and then these green stripes in the sequence are places where we've put in tags so that we can tell the wild type sequence from the reference, the wild type or reference sequence from the synthetic sequence. So we've kept the protein amino acids the same, but we've used genetic code redundancy to give a different DNA sequence. And we wanted to do those specifically inside the protein coding sequences. So you might think, oh, why are you going to change those? That's the protein. That's what's important. But the problem for us is that we really have no idea how gene regulation works on a large scale. And so we didn't want to put these watermarks in non-protein coding regions where we could be screwing up some regulatory signal. So all those changes happen inside protein coding regions. Then these green diamonds are places where we've put LOXP sites. And Tom told you how LOXP sites can cause recombinations during mitosis. So basically, any two of these diamonds during DNA synthesis as the cell is undergoing, sort of preparing for mitosis, they can pair up and you can get a deletion or an inversion or a translocation. You can also get copy number increase during G2 phase when there are two copies of the chromosomes inside the cell. This can also happen, and then you can get copy number increases. And then the X's show where we've deleted things. So here, I think, I don't know, some sort of transposon that was deleted from that place, another transposon deleted from that place. So that's the design on the computer. And then we made the DNA. Actually, for some of this, we had the DNA made for us, put it in as a circular back, and then sporulated, and then had cells that, so here it's the left arm of, so let's see, the left arm or right arm? So I guess so the right arm of the chromosome is being carried by a circular back. And since then, we've linearized the circular chromosome, and we're progressing to having whole chromosomes. But these sort of semi-synthetic chromosomes with one of the arms synthetic came out, I guess, last summer. All right. So now we've moved on to actually generating large pieces of DNA is still difficult. And it's sort of, each year it gets easier to make bigger and bigger things, but making a whole chromosome is still challenging. So one thing that we've moved to is a student course where we have students make the DNA for us. So here's a picture of the status of chromosome 3 at the end of the semester, where each of these little stripes is a little piece of DNA, about 700 base pairs or so, that we've given to a student. And then the greens are that the student has made perfect DNA, and the reds are that it's still in progress. Some areas seem a little more difficult than other areas, but still the students are doing a good job going through the whole chromosome, and then these little pieces are going on to the assembly stage. So hopefully we'll have chromosome 3 done shortly. This is just a schematic of Cre-LOXP recombination. And so since we need, I think acronym is a better acronym than scramble. So scrambling is essentially, I guess, the forerunner of acronym, because this is in yeast, and then it's going to be put into E. coli, I guess, microbial. So the acronym stands for, well, what it stands for is these LOXP sites recombine, and then you get genome rearrangements, genome scrambles. So synthetic chromosome rearrangement and modification by LOXP-mediated evolution. All right, so the goal is, so there are a couple of goals for this. One reason to do it is that there is a lot of interest in talk about minimal genomes. And minimal genomes are hard to design because we know what genes are essential one at a time, but we don't know requirements of genes too well. There's synthetic lethality experiments. There's people trying to map out the pairwise dependencies of the genome. So what pairs of genes can you delete at the same time? But then higher order combinations become very, very difficult to calculate. In fact, no one really knows how to calculate them. For example, think about something like generating amino acids. So you have an amino acid synthesis pathway and an amino acid import pathway. And if a cell is growing in rich medium, either it can import an amino acid from the outside or it can make it. But if it can't import it and can't make it, then it dies. So there's synthetic lethality between metabolic pathways for synthesis and metabolic pathways, and sort of not metabolic pathways, but more what people think about as a transport pathway. And so the logic of what you need at the same time is very confusing. So it's hard to think about what's minimal. But with a system like this, we can just start growing the cells, and then the cell will decide for us what's minimal by keep throwing things out of its genome until the cell dies, and then you back up one step and you say, oh, that's the cell that I want. But because we're growing these cells in a big population, we'll have enough living cells that we can go back and figure out what the local minima are. So we started doing that, and so this is characterization of some of these synthetic strains undergoing LOXP recombination. So these slides should show all the cells looking about the same. So there's the wild type cell, and then a synthetic cell where things haven't been triggered, a synthetic cell that has all the LOXP stuff, in it, but where the LOX-P recombination also hasn't been triggered, growing under different conditions, and the cells look the same. And then we turn on the LOX-P recombination, and the first 3 rows in each slide, in each panel, are essentially different controls. And the bottom row is the experimental strain. And so this is over time. So over time, as these experimental synthetic cells undergo more and more recombination, fewer and fewer cells are viable because a lot of cells have thrown out things that they need to live. But there are still viable cells. And then we did things like look at the viable cells and see whether they have any new metabolic requirements. We looked for lysine and methionine growth requirements because we knew that lysine and methionine sort of pathway genes were in this part of the chromosome. And so horizontally are different parts of the chromosome, and then each row is a different yeast strain. So, so these are yeast strains that have all lost parts of lysine synthesis. These strains have all lost methionine synthesis, and these strains have lost both. So now what we're doing is we're sort of going through these strains that have lost different parts of the genome and tuning up our analysis for when we have a whole genome that's undergoing LOX-P recombination. So with Beijing Genomics Institute, we've sequenced so far 76 strains on Illumina, done standard data filtering, looked at what's happened at the LOX-P sites, and then sort of a standard next-gen sequencing data flow where we map paired ends with bow tie. If we're able to map it, essentially it's sort of a null result that it matches the reference, or actually sort of the initial target. The ones that don't map, then we mask the LOX-P sites, we map the single ends, and then we figure out whether we have an insertion or an inversion or something else. And then we get these pictures that I guess you could call a scramblegram, where the yeast cells, so if you look at the LOX-P sites we put in, the LOX-P sites define 44 different segments. So each segment is between 2 LOX-P sites. And then for each segment, so here at the top is the control. We just color them in sort of a color scale. And then for each strain, you can sort of see what happened to all the segments. So, so here these gaps are deletions. And you see sort of there's a pattern of having a gap here and a pattern of having a gap here. So these are the strains that have the lysine and methionine growth requirements. And so these deletions are for lysine and methionine synthesis genes. So there are also big inversions. So here's a big inversion involving like a quarter to a third of the chromosome arm. And if there are only a couple changes, like 1 or 2 changes, you can sort of look at it by eye and say what happened. But once there are more than 2 or 3 changes, it's hard to figure out what exactly happened. And even sort of the smallest number of changes you need to get from the reference chromosome to one of these rearranged chromosomes. So here is a summary of the results looking at the types of changes we get in the synthetic genome when we scramble it. So the black, so each of these rows is a different strain, and then here are histograms of looking at the DNA, what's happened. So the black bar at the bottom is the number of segments that are sort of still in their reference orientation. So that's a segment that, say, even if it's been inverted, still if you look to the left and look to the right, its context is the same as the reference strain. Then the yellow is big pieces that we have deleted. The green are inversions. The blue are insertions, which are essentially sort of new, sort of someone has a new neighbor. So there's been a deletion. Now this one is the neighbor of this one. So something's been lost. So that sort of is scored as an inversion. And then the stuff that shows up above 44, or 43 or 44, the number of segments we had, is due to duplications. So that maybe in S2 phase, the DNA has been duplicated, and then these are actually a little more challenging to analyze if there's been a duplication, figuring out what's gone on, and so that's what we're working on now. So this is actually a classic problem. Before we had access, in the general sense, to DNA sequence data, people did phylogeny with gene order, and there's a lot of work figuring out minimum edit distances under inversion and deletion. And in fact, one of the people who did work on this is my brother, who did a linear time algorithm for minimum edit distance. And so sort of this is the next stage of analysis, is using minimum edit distance software or algorithms to figure out what was the process that led from this parent strain to these different strains, and see where there's sort of bottlenecks or critical points in genome evolution. And then what we're going to be applying that to is so this idea of minimal genomes, where we start with the wild type, and then in a bioreactor, the cell undergoes different gene loss, and then we'll get locally minimal genomes. So instead of trying to drive toward the global minimum, really in genome space it's more like local minima, and then we'll be able to look at the local minima and understand a lot more about gene function. So now a little bit about resources that we've generated for synthetic biology overall. So this is another sort of picture to put you on the axis of how sort of the size of the problem. So this axis is information content in megabases. So 1,000 megabase is a gigabase, so here's sort of a gigabase. And then on this axis is functional units in thousands. And we're plotting genomes where functional units are genes, books where functional units are paragraphs, and software functional units are let's see, I think they're procedures in the software release, so number of different subroutines. So Yeast over here has about the same information content as War and Peace or the Bible or the Bash shell. Then when we get to, so these are things that you can have definitely a single author for. So we've been writing multiple author tools, but definitely a single author for War and Peace. King James Bible, well, all right, so there are multiple authors there, but you know, but then when you get to more complicated systems like Drosophila, Arabidopsis, so they're about the same as the C compiler or the Linux kernel. So human is sort of also here, where you need to have a team of people who are working on the project. In America, I guess actually there's still a big debate on whether it was a team of people who designed people, or just one design or whatever. But luckily in Europe, it seems that you have evolved past those debates. All right, so BioStudio is our software that does computer-aided design, computer-aided manufacture to go from sort of an idea of what you want to make to so then multiple people can sort of edit different parts of it, so multiple people edit different parts of our genome. And then this sort of compiles down to oligos that then goes from electronic to physical, where then the oligos you order and you go through a manufacturing phase where you build the stuff. So Sarah Richardson sort of wrote our initial design software. Giovanni is working on this side and has taken over the design software. So Gene Design is out in the public. Actually, this whole system, I think it's on a CPAN repository, so you can download it and install it. Everything is open source, BSD. We start with a chromosome, go down in different phases to oligos, then back up. So one of the important things that we did is it took Jeff Buca months and months to come up with his first design for chromosome 9. Sarah with BioStudio, instead of months and months, took 5 minutes, and the design quality in terms of an objective cost function that we defined, it's optimal. And then you can just sort of see how much nicer it is in terms of restriction enzyme placement sites. So we're using these sites to recombine large pieces of DNA. They're supposed to be evenly spaced, so here the computer is able to space them evenly. Here with Jeff's design, they're sort of scattered very non-evenly. Then the manufacture phase, using students to do the manufacture, so I mentioned that earlier. And then, so it turns out that a lot of pieces of the system, of systems like this, you can sort of get off the shelf or someone else is working on it. So for example, people are making a lot of parts. We're making a lot of parts. So we were thinking about inventory control for our parts. And it turns out LBL JBay has made a very nice database for parts called the registry that you can use to sort of keep track of what you've made and where it is and stuff like that. But actually the workflow of how you go from idea to making the part, we've done a lot of looking and there are no good solutions for that we found. So we're writing a workflow system for us to use, and hopefully other people will be able to use it too, that we're going from oligos through assembly, so oligos to assembly to sort of further characterization, and so cues that people can go through and see. So it's just software engineering. I shouldn't say just. It's a lot of work, and but it's important. So to learn about this, I actually ran some gels myself. So these were my first experience in 23 years, and they worked. Clone to well mapping, some more workflow. So as I was doing this project, one of the things that we had to do was to So you make DNA, and then you sequence it, and then match your sequence with the sequence you wanted and see if there are any errors. So you sort of go blind doing this. And when I was working in the lab to figure out how synthetic biology worked, so instead of doing that process, I wrote the engine that we're using now to do to do sequence error checking, and then Pablo Lee, an undergraduate, wrapped it in a nice Ruby on Rails interface. So then all this is being shared, BSD license, and then underneath we're using entirely free tools. So actually the authentication now is through your Google account. So instead of writing our own authentication system, it goes through Google. Anyway, and if anyone's interested in a system like that, you're welcome to talk to me. We're dedicated to sharing it openly and making no money from it. So here's our iGEM team's project this past year, was making bread with vitamin A synthesis. So actually the bread and the lab sort of smell like carrots. So I didn't realize that the vitamin A synthesis pathway, the intermediates have a carrot-like smell, but they do. So, so nice orange bread. It's going through IRB approval now to figure out whether people can eat it. Although I think some of the people might have eaten it already, but without telling our ethics officers, but they weren't allowed to. And then here's obviously the bread maker. Right, so I'll skip that. So now, so Reanimator is the name of, so I don't know if you know this movie, but it was, it was a very formative movie in graduate school. And so now there are these projects to, so we're making yeast, but obviously, so one of the goals is to make other organisms. So a lot of cool interest in, so the mammoth project to reanimate the mammoth, dodo, dinosaur. So ancestral yeast is one of the targets that we'll probably go for, so maybe pre-duplication yeast. So that project would be $12 million now, but then if you think about, so $12 million now, so that's about the cost of an attack ad in our presidential elections. So, so these more complex species, $1 billion now, but in 2020, based on Moore's law, 10 years, 100 times cheaper, only $10 million. So it'll be really feasible to start doing this. The challenge is that you're going to have to sort of walk backwards in the tree of life to a common ancestor and then back out in order for, say, transcription factors that are encoded in the DNA to have, or regulatory elements encoded in the DNA to have the right transcription factor proteins around. So it's not as if you made dinosaur DNA now, what would you do with it? There's no host cell you could put it into that would start the system running right. So it's actually going to be a challenge, but it's sort of a feasible challenge, and it's sort of really going to be cool, and it seems to me like other than doing deep sequencing of fossils to, whether there's any DNA left, but it is sort of really feasible and sort of cool, and the way things are going, financially, it's not really going to be so much of a big deal. So, so now again, I have to thank Jeff Buca and the people who've worked on the project. Carlos Uruguayan is a visionary guy from Autodesk who's been helping us out a lot with resources. And actually one of our students is at Autodesk Software now, helping plug our things into their software system. And then again, our collaborators and funding, and thank you.